So yeah, thanks for joining us today, Jason. So yeah, I'm Nico, and this is the Method Made podcast. And here we have been discussing the sort of operational challenges in technology transfer, starting off broadly speaking, and most recently in cell and gene therapy. And today I would love to talk to you about some of the unique standardization and potentially documentation challenges that cell and gene therapy faces. And to kick things off, what I see as unique about cell and gene therapy is that it's a personalized medicine. So when you're manufacturing vaccines or small molecules, you can make a huge batch at once and you can treat a bunch of people all at the same time. And cell and gene therapy is different in that it is personalized and one patient's cells are manipulated and given directly back to them. And I know that Ori system has some controls that facilitate some of these challenges. So maybe to start with, a good question would be, what do you see as the sort of largest standardization and documentation challenges in cell and gene today?
Yeah, I mean, I think you certainly nailed the differences on the head to say we have a order of magnitude more batches in cell therapy or gene therapy than we do with any other modality. As you do with biologics, as you said, small molecules, vaccines, we make a generic kind of small g product that is applicable to any patient anywhere, and they make millions of doses at a time. We make one dose for one patient. And so that increases by an order of magnitude all the paperwork, all the batch records, all the quality QC effort, the release processes. And that really has consumed the energy and the time and a lot of investment from the industry to try and, as best they can, of course, we're ensuring quality, but ensure it in a way that makes sense rather than just paper batch records that's 1,000 pages. I've seen a paper batch record for a cell therapy with 16 3-ring binders, over 1,000 pages stacked up, and you have two people that have to review that and sign off every single page. That takes time. That is very inefficient. And then most of the industry today is still on paper. So that's paper batch records, paper lab notebooks. And so that just multiplies as you get to the hundreds and then the thousands of doses. So you can imagine how much paper that generates and how inefficient that is. So I think the move to fully digital that is enabled by next generation systems, might be MES systems, LIMS. We're partnered with Benchling on the ELN side, with TrackCell and others on the COI, COC side. So there's some players that are digitizing various pieces of the value chain, which will help. And this means true digital, not paper on glass. A lot of times when people say, oh, we're digital, there's no paper or cleaner, what they're doing is they're uploading PDF files into a batch record. That's not really what we mean.
Yeah. And absolutely. I didn't know that's a huge volume of paperwork. And so something else that I see is unique with cell and gene is the time constraints are also different. So with the COVID vaccines, we were all patiently waiting. There was, of course, damage that happened, but a lot of us were patiently waiting at home for that text message when they were ready. But with cell and gene, these are usually happening in not the best of circumstances that people are getting these treatments. And yeah, I can imagine that these binders full of paperwork are standing before patients are getting their treatments.
Absolutely.
Yeah, what's your perspective on that? Is that kind of like a bottleneck in the process?
Yeah, absolutely. It delays or lengthens vein to vein time. The key bottleneck today, I mean, by the theory of constraints, you can only have one bottleneck. You can't have two.
Right.
So you have one constraint, that is today manufacturing. It's really the expansion phase of the cells getting from the patient cells that have been genetically reprogrammed to whatever your therapeutic dose is, often takes 5 to 7 days or even longer. So that is a core bottleneck we have to fix. And that's really where the Ori technology is focused on, fixing that bottleneck that really hinders the throughput of these processes around activation, transduction, expansion, and harvest. Those are the core bottleneck. But just as you highlighted, as soon as you eliminate that bottleneck and increase the throughput, the next bottleneck appears, and that's at QA and QC at the end of the process. So it definitely acts as a process limiter, throughput limiter at the next generation, but also just one that lengthens the amount of time. People talk about 2 weeks to do a sterility test. And that's two more weeks the patient has to wait until they get their therapy. A lot of times, if patients in an acute scenario, they'll ship the product at risk. They may even infuse it at risk. because if we find a contamination, we're just going to treat the patient with antibiotics anyway. So rather than letting them die because the therapeutic is contaminated, it's a lot of suboptimal processes because we don't have the tools we need. We don't have the digital platforms. We don't have the automation tools. We don't have rapid sterility testing. We don't have automated QA and QC. So these are all challenges that we're actively engaged in solving with our customers and our partners.
Okay, that is great to know. So you mentioned quality control and working with the best of what's available currently. And yeah, I'm curious if there are ever situations where these quality processes as well established in cell and gene as they are in more mature technologies, or are we still in the learning phase of what quality means in this industry?
The standards are certainly the same. GMP requires us to uphold those standards. What is lacking are the tools. So the tools that we're using were built for another generation, 10 or 20 years old of medicine. So it's the same tools we're using in biologics, or the same tools we're using in other areas where, as we said, we're producing hundreds of thousands or millions of doses. And oftentimes the patient isn't in as acute need as they are in cell and gene. So I think it's really the tools that aren't fit for purpose. Yeah. We're using, as I said, sterility testing that takes 14 days. Why? Why does it take so long? We're talking now about 24 or 48 hour rapid sterility testing that's required for this field and for this industry. So I think innovation is required really to try and bring, just as we brought the Eero system into the manufacturing workflow, which is a new innovation replacing 10-year-old technologies. We need the same to happen upstream and downstream in the value chain, particularly around QA and QC. And one of the things that we're looking to work with some of the leading providers in things like flow cytometry or cell counting or sterility, really to connect the data flow and also to connect the fluid handling. So you don't have a person there who has to do all the sample prep that takes hours and for multiple samples every time. And if we can automate some of those, like let's call it the low-hanging fruit, we don't have to totally reinvent flow cytometry. Potentially, you can just make it better, faster, cheaper. And that's where we're focusing our attention in the near term anyway.
Okay, so yeah, I think it'd be great to discuss how Ori system fits into this. And one thing that I think would be a great sort of segue into there is talking about the sort of manufacturing workflow and the differences from traditional manufacturing systems. So in more traditional biotech, you've got CROs that will develop a new technology, pass them over to CDMOs to do process development and scale up, and then there'll be manufacturing. And I've heard that in cell and gene, there are sort of emerging manufacturing on sites in hospitals. So I'd love to hear, where is the Ori system actually being used? Is it at these separate CDMOs, or samples getting flown out to them and then back to hospitals? Yeah, how is that actually happening?
So the Eero system is really targeted to 4 different types of customers. So there's therapy developers, both big and small. The big pharma guys like a Novartis or a J&J or a BMS has different needs than a small CGT startup that doesn't have any products. So obviously we treat those as 2 different customers because they have different needs, but they're both developing therapies. And so our technology goes into their labs, maybe their R&D lab, their PD lab, maybe into their clinical manufacturing, and then ultimately commercial manufacturing. That's one use case that the technology is very well designed for. The second would be with a CDMO who's servicing many customers, doing some of those services on their behalf. So oftentimes they want to get IND ready, to get their sort of regulatory filing in. They might do clinical manufacturing on behalf of that customer, but they're really the process experts. They understand what tools to bring to bear, and they have the knowledge and expertise to do that in a cost-effective and time-effective manner. So we've just announced our preferred partner network, which is a network of some of the leading CDMOs in the world. People like Elevate Bio, Charles River, CTMC, Kincell. These are folks that really understand the development process and can get products in the hands of patients, or in the hands of clinicians, to deliver to patients very quickly. So that kind of second bucket, you've got therapy developers is the first, CDMOs is the second, and then academic research centers is the third. AMCs are developing the next generation of therapy. So the tool that we've built, the Eero system, is flexible enough to do research on. So the idea was to build a tool that they can use at the research scale that scales seamlessly into clinical and then commercial scale manufacturing without the need to do comparability or to change your process fundamentally multiple times, which today is the reality. And it's why it takes so long, and it's why it's so expensive, is because they often come out of academia in tea flasks or bags, some manual tool that's been around for generations that is definitely not fit for scale, but it's fine and it's cheap and it's relatively easy to use, but it will never get you a commercially viable product. It's that, what's the sweet spot? A lot of the conversations I've had over the last several weeks is, what is the sweet spot to try and change a process? The best thing is to start out on a flexible platform that you never have to change. If it works, you don't ever have to change it. That's the best scenario. If you've already got a process that is preclinical, kind of PD, translation PD, that's a great time because you're going to be doing process changes anyway. And so introducing automation at that stage while you're optimizing is optimal, but certainly no later than phase 1-2. Early clinical is the last chance you're going to have to really make final process changes and to get your process honed so that you have a reasonable cost of goods. You've got a target throughput that'll get you the number of patients you want. You've got the safety and efficacy profile that you need. And really phase 1-2 is the last chance you have to do that. Once you get into pivotal, it's too late to make those changes. And so that's really where we're seeing the most utility and the most interest around the Eero platform.
And that makes a lot of sense. What I've heard is that at that point, once you're within those clinical trials, you'd have to repeat so much of that paperwork if you were to make any process changes. So basically no one is willing to do it. And it's so interesting to hear.
It's not actually true in the early phase of clinical, because when you're doing phase 1-2, you've just started the process. You haven't done your pivotal trial. You haven't locked your process most often. So you do have time in that early clinical phase, phase 1-2, but certainly not after that.
Okay. So that's the cutoff point there.
Yeah, that's the latest.
So one thing you mentioned that the system is flexible and that it can be used at all three, the discovery, development, and manufacturing scales. I'd love to talk about the challenges of technology transfer, because this has been just a constant conversation point over the past year, is how do we get, as you mentioned, from shake flasks to something that is actually manufacturable? And so one thing that's come up is there are often different documentation expectations and standardization and standards between discovery labs and engineers. The engineers often need parameters. There are higher documentation standards, but it's a lot of it also, my understanding in the process development work, is trying to equate a system which isn't very well controlled, like a shake flask, and translating that into a bioreactor, which has process analytical technology and all of that. So I'd love to hear about how Ori System can facilitate that technology transfer. What does that mean that you can use the same system at discovery and then into process development and manufacturing? Like, how does that change things?
Just as you said, oftentimes these have been siloed activities. Your research team may not even talk to your development team, which definitely doesn't talk to your translation team, which definitely doesn't talk to your process development team, who definitely doesn't talk to clinical manufacturing. So there's 4 or 5 different teams that are involved before you're sort of really at clinical scale, and process transfer will have to happen throughout those different handoff points. And they each have different focus areas and different KPIs, and that causes a lot of disjointed products to get developed and a lot of rework to have to be done. I think more and more we're seeing those preclinical process elements or those preclinical silos being broken down so that you have more communication, more shared vision. We're talking about putting things like target COGS, target throughput in your TPP, so we know what we're shooting at, not just clinical endpoints, not just safety and efficacy endpoints, but things that are ultimately going to lead to a commercially viable product that need to be understood and need to be managed early in the process. So what that really means from the perspective of developers using automation technology is that in the past, they didn't choose automation technology early because it was too inflexible and seemed to be too expensive. And so before you have a process, before you're certain what the details are, I just wanted to have manual things that I could move around the lab myself. Now, because we've got more flexible tools, you're not constrained by the ability of the limitations of existing technology. And the Eero is really built for that purpose, to be helping researchers tinkering around in the labs, changing this parameter, that parameter, testing, how does that work, and really be able to do that in a fast way. And so what that means as far as time to clinic, which is one of the key measurements that people think about, we think we can shave about 1.5 years off of the time to clinic, which is a massive uplift for a young company. Time is money in drug development. So every day you take, or every week or every month, costs more and more money. We know the funding challenges right now that are out there in the world. Tech transfer is one of those things that takes a long time. And especially if it's on paper, a lot of it is a training burden. You have people, and we're trying to teach a new set of people at a different place, at a different site, or an additional site, how to do that process, as well as the people who invented it. And that often takes a lot of training. There's a lot of documentation. You have to sign and say, I trained and I ticked and I know what to do. And then, of course, people only understood about half or 2/3 of what they had to do. And so you get a lot of variability. There's retraining. All those things contribute to this very kind of long timeline, 10 plus years of developing a cell therapy. In a proof of concept that we did with the folks down at MD Anderson and CTMC, we tech transferred a process that we had put onto the system. So we'd taken it off of its existing Flask technology, put it on the Eero system. We did that in 6 weeks. That is not easy to do in 6 weeks, but it was very quick because of the adaptive nature of the system. Maybe even more impressively, we took that process from Houston and tech transferred it to Philadelphia in 2 weeks. Doing tech transfer in 2 weeks, this normally takes a year, 9 to 12 months to do that well. But because we've taken 60% of the people out of the process, effectively tech transfer is pushing a button, having the protocol transferred to another Eero system that's in a different location. It still knows what to do. It still does it exactly the same way. That's the benefit of automation. It doesn't have a brain of its own. It gets told what to do and it does it. And so it eliminates most of the training burden and a lot of that people training and the validation to say, okay, we were doing it this way. Now we can validate that we're doing it the same way here. So it dramatically cuts tech transfer time. And throughout the development of it, it's now a 10-year process. You would probably tech transfer 2 or 3 times, maybe out of an academic lab into a company, maybe out of a company into a CDMO, maybe out of a CDMO into a manufacturing facility or a larger CDMO. Every time you do that, it's a year or 2 years and a half. So we estimate that based on just on shortening tech transfer time alone, not the benefits of faster optimization, but just by shortening tech transfer time, we can shave 3 years off the drug development timeline for a cell therapy. 3 years out of 10 is massive for patients, massive for sponsors, and that's all benefits of just more efficient path through the system.
That's very impressive. So in tech transfer, I see 2 components here. So what you guys are doing is standardizing the hardware such that there's less confusion because it's the same system that's being operated in one place to another. You also mentioned that there's still this challenge of training and retraining from someone at one facility that's developed a culturing procedure to someone at another. Recently, we spoke with some academic researchers working with these types of cells, and it sounds like it is quite difficult to effectively document all these procedures. So I'm curious if you see, aside from the hardware as a way, standardizing the hardware as a way to accelerate the tech transfer, do you see that there is still opportunity for improvement in that communication of how to actually use it in those sense?
Absolutely. I mean, I think we talk a lot about the talent gap in cell therapy. You need lots of educated, talented people who understand how to do these pieces. And there just aren't enough of them. And there aren't enough of them in all the places you need them because there's concentration of talent in places like Boston or Philadelphia or Houston or Amsterdam or elsewhere, but there aren't where necessarily in every place you need them. We find the kind of the digitizing aspect of this. So we're able to generate, ours is a digital cloud native platform. We generate data and have real-time monitoring. We had a partner who was in San Francisco that was running a parallel process with one in the Eero and one in a competing technology, trying to validate that they could transfer off one onto the Eero. The system flagged an anomaly. They said this reading is below where it should be at this stage in the process. And so we looked at it and we said, this looks like there may be a contamination. We called the partner and said, this was early. They were in San Francisco, so it was early for them. And so when you get in, you should sample both cultures and see what we think there's a contamination. And it turned out that there was a contamination both in the Eero system and in the Prodigy system they were comparing it against. And it had been that an operator upstream had contaminated the starting material, but they wouldn't have known that in the Prodigy because it's not cloud enabled. So they would have gone through the process and then at the very end, they would have tested it and they would have found that it was contaminated. So I think the adding digital element, both to the training upfront component, the documentation component, but also just the process itself, it powers so much more information and ability to intervene as needed. The system is built on the back of some machine learning and AI algorithms such that it learns. So something happens, and then it learns when the operator makes a correction, and it says, "Oh, okay, that's what's supposed to happen then." And once it see that happen 100 times and then 1,000 times, the system learns what to do. This is the whole kind of idea of machine learning, but the idea that we could potentially not only tech transfer more quickly for those people that are left in the process, we could de-skill them so that there's much more human capital available. You don't have to be a PhD immunologist. You could be a high school graduate and still run these kinds of processes with the help of automated tools like the Eero, maybe with the help of better training and better digital tools as well. So I think there's a need to really apply innovation across the value chain here, even for those things where we don't view this as replacing a bunch of people. There's a massive gap of the skilled labor we need that actually opens up the labor pool quite significantly, but we're still going to have people in the process. There's still going to be people doing QA and QC or warehousing or upfront chain of custody, or there's going to be people in the process. So knowing that those people are well trained, they know what to do, they know exactly, they can replicate it no matter what site in the world they're in. These are all very important things to create standards around and make sure that we get ultimately the goal is to get the same safe and effective product out the other side of a process.
Yeah, it makes a lot of sense. And something that keeps coming up is sometimes there is this skill gap, and in technology transfers, this seems to be one of the communication challenges, is what some people describe as like the curse of knowledge, that the expert who has spent a PhD, for example, developing a process takes things for granted, ways that they might handle cells and that type of thing, and assume that someone else that's going to be operating a, say, manufacturing or production system is going to have the same level of, say, troubleshooting, like off the cuff troubleshooting abilities. And that's not always the case. Sometimes there is that skill gap. So it's amazing to hear that through automation, one of the opportunities here is to reduce the need for that off-the-cuff troubleshooting, as you said.
Yeah, I don't know that we want to turn everybody into a cyborg, but having people that are supplemented by technology that helps them do their job more efficiently, better. You also have that, call it the curse of knowledge or something similar. You have a very real risk of that kind of tribal knowledge walking out the door, right? The average retention rate for an employee in a clean room in cell therapy manufacturing is less than a year. Because oftentimes they're a grade B, they're in a bunny suit, goggles, 2 pairs of gloves. Like, it's just not that pleasant an environment to work in. And especially if you did your PhD in immunology, that probably isn't where you expected to be spending your time. That having the ability to standardize, to automate, to digitize where appropriate, that knowledge and make sure it's available for the next generation of operators or whomever is critical, I think.
And so that's one form of risk that these companies face, from what I've heard, is if someone walks out the door, that's a huge company risk. If there has been this type of tribal knowledge, regardless of how simple your process is, if it isn't replicated the same way, that's going to cause you a lot of problems. Yeah.
100%. I know we were joking about that kind of YouTube video about making a peanut butter and jelly sandwich. It's not as easy as making a peanut butter and jelly sandwich, but there's also a way to make a peanut butter and jelly sandwich. Yeah, no, absolutely. It's not as easy or straightforward as we think it might be.
Wow. Okay. Yeah, this has been super informative. I guess one last question that'd be great to kick it off is, we talked a little bit about bottlenecks. I'd love to hear, so after manufacturing and cell and gene therapy, what do you see as the emerging next sort of challenge frontiers in the industry?
If you think about the black box of manufacturing, which includes, I hear a lot about apheresis beds, not having enough space to get the apheresis for the starting material as being a bottleneck, because those are being used by other patients who sometimes might be more acute. If you're an autoimmune patient, you might get bumped for a cancer patient or whatever. So that's one of the areas that I hear quite a lot about, is like our actual physical ability to treat and provide treatment for patients. And most of the cell and gene therapy these days is concentrated in specialist centers that have, a lot of infrastructure and have the ability to deal with very sick people and have knowledge about how to administer these therapies. But for many of them, many patients, it involves picking up your family and moving for 6 months. Not every family can afford to do that. Many of them have jobs and they have to continue to continue health care. So this idea of moving treatment into the community, I think, is a massive opportunity to alleviate some of the bottlenecks, either on the front end, but also just on treatment access in general and prescribing. At the end of the day, these are expensive products. if we can fix manufacturing, drive down cost of goods, we're seeing already some of these products being second line when they used to be fourth line. So we've seen Breyanzi and Carvykti in particular double in volume year over year after they got second line designation. So there's more and more patients that need the help or that need these interventions, but we need to be able to deliver them in more standardized, more accessible settings like community-based hospitals. And so that's where things like distributed manufacturing may come in. It's where other things like being able to manage things like CRS and the education of those community centers where they may not treat 10 CAR T patients a month. They may only treat one per quarter. So how do you make sure that they do it right? So some of these barriers I think will help also that are outside the manufacturing suite, but will help us get broader coverage as we're able to get better access for patients. We'll need better provision in all the ways that means, both on the front end of the process, but also on the back end.
Wow. Okay. Yeah. What an informative discussion here, where we're at and where we're going with this industry. So thank you so much for joining us today.